Hello students, in our previous video we discussed about lambda Phage DNA and the various regulatory factors involved in channelizing into either a lytic cycle or lysogeny cycle. So in this video we will try to understand how the Phage vectors are designed. So before that I will again go through some of the finer details. Now this is the whole length of lambda DNA. So now that is made out of 48,502 base pair. We call this is the 100% genome size, and at the end we have a cos site which is 12 base pair overhang, because this consists of about 40 genes, and the significance of these 40 genes, they are all arranged in the form of a functional cluster. On the left hand side we have the genes responsible for the protein assembly of head and tail. Now in the center we have the regulatory elements required for the lysogeny, and in the right hand side we have the regulatory elements required for the lytic cycle. Now let's see what kind of strategies they have employed for the construction of Phage vectors. So these particular numbers are very important. Now the genome size is 48,502 base pair, or we can say 48.5 kilobase, which is 100%. And I told you that for the packaging of this DNA, the size is very important. So it can accommodate additional 5% DNA. Now that additional 5% DNA, so that makes from 100% to 105%. So this can accommodate an additional 2,425 bases. That will come to 50.9 kilobases. Alright. Then it also has a minimum size. Suppose the minimum size for packaging is 75% of the genome size, so which is 36.3 kilobase, or I would say that it can forego 12,125 DNA bases. It can accommodate this many bases, and it can also forego this many bases. Anything less than this, or anything less than 36.3 kilobase, then it will not going to get packaged. So this information is very important, basically because now this is going to offer us what is known as a size selection, especially in case of a clone identification. In case of a plasmid, the identification of the clone is through antibiotic resistant gene. But here the clones are going to be identified merely based on the size of the vector DNA or the clone DNA. Now when you look into the wild type, so wild type for sure do not encourage, so that it can be a good candidate for the generation of vectors. So what they have done is that, so now for the purpose of recombinant DNA technology, any vector what we have designed should get into the lytic cycle but not the lysogeny cycle. So as a result of that, so because there is a size of it can accommodate 105%, and it can also like a minimum 75% of DNA is required. So they realize that why can't we remove this particular lysogeny regulatory elements. So in other words, so from J to N, because these particular DNA segments are not important. So apart from that, even there are certain unwanted DNA segments in this region, as well as the unwanted segment in this particular region. So if you remove all those things, so then we can easily accommodate an insertion size of 24.6 kilobase. So initially it was very bleak, so that we can only accommodate 2,425 bases. Now when we look into the strategies, what is required for a Phage vector, we are not interested in this particular DNA segment. So can we remove this DNA segment and replacing this DNA segment with the DNA of our choice? That was the strategy. So while calculating the whole process, we found out that an insertion size of as big as 24.6 kilobase can be easily inserted into this size. So as a result of that, wild type for sure no, but when you are going to design Phage vectors, then we can include 24,600 base pair. The moment we remove this, so we will end up in having a region. So what we call this as a left arm of the vector, and this particular region is called as the right arm of the vector, because this 12 base pair cos site is very, very important for the packaging, isn't it? So we have a left arm and we have a right arm, and this left arm and right arm put together, this will amount to 23,902 base pair or 23.9 kilobase. So now, can these two arms join together as a vector? Can it going to get packaged? Definitely not, because the minimum size required is 36,376 base pair or 36.3 kilobase. So now this vector with left arm and right arm cannot package. So if you want to have these two things to get packaged, we have to insert a DNA, so that that length of the DNA should come between 75% and 100%. So that is the space we have it, so that we can somewhere incorporate between 75 to 105. So during this, so we can insert as big as 24,600 base pairs. So now you can understand that study of lysogeny cycle and lytic cycle is very important while designing the whole thing. I told you, like, this segment and this segment has lot of restriction site. All those restriction sites has to be modified so that they are not going to be functional. Now that one is easily carried out by the point mutations, substitutions, deletions, because these particular techniques are mastered during that particular time. So most of the restriction sites which are present in good numbers are all mutated so that they are not going to be functional anymore. That one they did it using point mutations, substitutions, deletions, and in the later stages they also incorporated some synthetic oligonucleotide so that they can create a critical restriction site. The common restriction site which are used in case of a Phage vectors, they are EcoR1, HinD3, SAL1 as well as SAC1. So now you got an idea that how Phage vectors are designed. Phage vectors have a left arm, then they do have a right arm, because we have removed the DNA segments and other unwanted DNA segments responsible for the lysogeny. So then the whole DNA becomes very short. That particular short DNA cannot be packaged because there is a size selection. 75% of DNA with two cos sites are very important. So in order to make it suitable for packaging, then we have to insert our foreign DNA. Now that foreign DNA is the DNA of our choice, which we are going to incorporate into these vectors. Now by looking into all these things, so in the Phage vectors, so we can include or we can insert as big as 24.6 kilobase DNA. Now can you imagine that in case of a plasmid vector, maximum size we can incorporate is only 6 kilobase. Now in these Phage vectors, we have introducing 24.6 kilobase or 24,600 base pair. Now we will see what type of vectors we are going to have it. So now with Phage vectors, we have two kinds. One is insertional vectors, another one is replacement vectors. The difference between insertional and replacement vectors is that now here we have a left arm and here we have the right arm. Then these two things are linked with an unique restriction site. All right. Now what we do that we are going to cleave. We are using a restriction enzyme. We get a left arm, we get a right arm. Then this is the DNA of our choice. Then we will going to perform the ligation. So now the question is, we are going to get these two arms joined together. Now that is the vector we are going to generate it back, but we are also going to get a clone. Okay, so now here, so now we are going to insert the DNA at a unique site. So because of that, we call these as an insertional vectors. Now in case of a replacement vectors, again we have a left arm, we have a right arm. So instead of a unique site, I think we are going to have the region which is responsible for the lysogeny segments are replaced with an unknown DNA segment. So what we call this as a stuffer fragment. Alright. Now during the cloning, what we have done is that, so we take these replacement vectors which have this stuffer fragment. Now that particular one is treated with restriction enzymes that results in the generation of a left arm, right arm, and the stuffer fragment is completely hydrolyzed because of the restriction endonuclease activity. Then this arm together with the DNA of our choice, we will subject for the process of ligation. Again, like here, so there is a possibility of this arm and this arm fused together to get the vector back without the stuffer fragment. Plus, there is a possibility of clone. Now, these vectors are not going to be incorporated because it is lesser than what is the minimum requirement, that is the 75%. So now the moment we insert, that means once we insert the DNA because of the clone, the size is going to be appropriate, which lies between 75% and 100%. It is going to be packaged. So as a result, now the size selection is critical for the identification of the clone. So now we have the insertional vectors, now we have the replacement vectors. So now using this mechanism, so we have mainly two types of vectors, that is the Phage vectors. First one is called as a Caron vectors. So in case of a Caron vectors, they have used the help of LAGZ DNA, which will code for the beta galactosidase. We all know that LAGZ DNA, the presence of LAGZ DNA, so now this offers a clonal selection based on the chromogenic method. So here, so in the presence of an IPTG and XGAL, because of this activity, now in case of a Phage, we don't get the colony, but we are going to get the plaques. So I'm going to describe what exactly plaques are. So we are going to get a blue colored plaque. Now here, in case of a Caron vectors, in case of a insertional vectors, in the unique site we have the LAGZ DNA. So just enough is required for that. So now what happens? In this LAGZ DNA, we are going to insert the DNA of our choice. The moment we insert this particular DNA into the LAGZ DNA, now the LAGZ DNA loses its function. As a result, in the presence of XGAL and IPTG, we don't get any blue colored plaque in fact. So if you do get a blue colored plaque, then that is not a clone. But if you do get a white plaque, so now that really selects that, okay, that is going to be the clone. Now that is the insertional vector. In case of a replacement vector, what the Caron vectors uses that, so in the stuffer fragment, so we are going to have a full length LAGZ DNA. So now this LAGZ DNA is going to be replaced by the DNA of our choice. So so in case of a Caron vectors, whether it is insertional vectors or replacement vector, LAGZ DNA is used for the clonal selection. Now another group also developed one more set of vectors. They called as lambda GT10 vectors. Now these lambda GT10 vectors, instead of a LAGZ DNA, they are going to use the C1 repressor gene. We know the significance of the C1 repressor gene because C1 repressor gene is very important, that it will have the lambda DNA to get into the lysogeny cycle. So I told you that the main purpose of our vectors is to get into the lytic cycle. So now suppose, so this C1 repressor gene is there in the unique site or it is there here in the stuffer fragment. So the moment we insert the DNA of our choice in the C1 repressor gene, then the C1 repressor gene is no more functional. So as a result, this will never enter into the lysogeny cycle. It will always enter into the lytic cycle. Whereas in case of a C1 repressor gene in the replacement vectors, it is present as a stuffer fragment. It is completely eliminated. Then we are going to get the clone. So now we discussed like what are insertional vectors and what are replacement vectors. and how the Karan vectors are designed. So in case of the Karan vectors, they have used laxi DNA for their clonal selection. In case of the lambda G10 vectors, they have used C1 repressor gene for the clonal selection. We have one more critical selection. So you have to be a little bit, it's a very tricky one. So it is called as a SPI plus and SPI minus phenotype. So now again, this SPI plus and SPI minus phenotype are used for the clonal selection in special type of vectors. Okay, we will try to understand what is the SPI plus phenotype and SPI minus phenotype. Alright, now this is an E. coli. Okay, now this E. coli do consisting of genomic DNA. Now a particular strain, E. coli strain, is infected with a P2 bacteriophage. I think in the initial one I have mentioned that these P2 bacteriophage and then the C bacteriophage series, they all resemble lambda DNA, isn't it? They all have a similar type of lysogeny and the lytic cycle. So when you do have this E. coli strain initially infected with P2 bacteriophage, now the DNA which is inserted from this P2 bacteriophage, invariably it will get into the lysogeny. So this will get incorporated into the host genomic DNA. So now this is the host, the P2 bacteriophage in the lysogeny form. Alright. Now this particular form is called as a P2 lysogeny. This is also called as a prophage phase. Okay. Now I will repeat one more time. We have the E. coli strain. Initially it is infected with P2 bacteriophage. P2 bacteriophage invariably enters into the lysogeny cycle rather than a lytic cycle. So we all know what exactly lysogeny means. Now this particular the DNA of phage get incorporated into the host genomic DNA. Now this is what is called. Now this particular condition of a host, we call this as P2 lysogeny or prophage. Now you take this particular host where we have the P2 bacteriophage DNA in the lysogeny. So now in this P2 lysogeny host, when you infect with regular lambda DNA, this will unable to infect this P2 lysogeny because this is unable to do that one. Now this particular phenotype is called as an SPI plus, meaning sensitive to P2 inhibition, meaning that because this has got a P2 lambda DNA in the lysogeny form with the host genomic DNA, now this particular lambda DNA is unable to infect. Okay, it will not get into any lysogeny cycle or a lytic cycle. Now that particular condition is called as a sensitive to P2 inhibition or SPI plus, or it is an SPI plus phenotype. They were trying to understand why the lambda DNA unable to carry out lysogeny or a lytic cycle in a host in which P2 bacteriophage DNA is in the lysogeny form. Then on a detailed analysis, they found out that this lambda DNA carries a functional gene RED and GAM gene. Usually these two are part of lysogeny cluster of lambda DNA. Alright, so because of this GAM and RED gene, the lambda DNA is unable to infect in the P2 lysogeny host. Now what they have done is that they removed this RED and GAM gene from the lambda DNA. When they repeated this experiment, now in the absence of RED and GAM gene, now the lambda DNA will infect the P2 lysogeny host to the lytic cycle. Now that phenotype is called as an SPI minus, meaning no more sensitive to P2 inhibition. So now between this SPI plus and SPI minus phenotype, the critical element involved is the RED and GAM gene, which is present in the lysogeny cluster of a lambda DNA. So what they have done is that, okay, using this RED and GAM gene, so they have created and another type of vectors. So now those vectors are called as the lambda 2001, lambda dash and EMBL series. What they have done is that, so they have taken the left arm and the right arm of the vector. Now we know that we also have a stuffer fragment in the replacement vectors. In the stuffer fragment, so they have included red and gam gene. Okay, so now in case of a red and gam gene, so now if you try to have the vector infected into the P2 lysogeny host, this will never get into the lytic cycle. The moment we remove this red and gam gene from the stuffer fragment, then it is easy to incorporate the DNA of our choice, so in a P2 lysogeny. So now again here the red and gam gene are used for the clonal identification. Again, for this clonal identification, it has to be done only in the P2 lysogeny or in the prophage phase, not in a regular E. coli. In case of a regular E. coli, now using this particular strategy, it is difficult to identify the whole process, whereas this can be easily done using the P2 lysogeny host. So now we have discussed that, okay, we have the Caron type of vectors, we have the lambda GT10 vectors, where in case of Caron they have used LAGZ DNA, in case of lambda G10 series, so we have used C1 repressor gene. Then we have this series, that is the lambda 2001 lambda dash EMBL series. So it is based on the SPI plus and SPI minus phenotype. So here the host happens to be the P2 lysogeny host, and the selection is through the incorporation of red and gam gene in the stuffer fragment. So in all these vectors, so we are going to have, after the ligation, left arm and the right arm, they can combine together to form the vector, or we can get the vector back. Another one is going to be the clone. So now when you look into these two, either the vector or the clone, how are we going to incorporate this into the E. coli host? We know that larger the DNA segment, especially more than 10 kilobase, it is difficult to transfect the DNA into the E. coli host. So now how this big DNA, either the vector or the clone, we are going to incorporate in the process of a transfection, we will discuss that in the next video. Thank you very much.